Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Today we're going to discuss a paper titled Polarity Orientation of Microtubules in Hippocampal Neurons, Uniformity in the Axon and Non-Uniformity in the Dendrites. It's by Peter Baas and colleagues, published in PNAS in 1988. Before we get into the paper, I'd like to start by talking a little bit about microtubules. You all know that microtubules are part of the cytoskeleton within the cell, and they're made up of individual monomers, proteins that is, that are called tubulin monomers. And I like to think of it a little bit like Legos. I mean, tubulin, which is a protein, so it's a gene product, has a polarity. That is, this side of the tubulin monomer is different than this side of the monomer. And it turns out that the tubulin monomers, they say polymerize, which means that they kind of grow in length to form protofilaments, which are kind of just long stretches of monomers like this. So many copies of the tubulin monomers like this, and of course it can go in this direction too. And to form a microtubule in mammals, there are 13 protofilaments. And if you look at a microtubule in cross-section, then you see something like this. Let's see if I can draw this. You can see this ring of protofilaments, and it's a tube. It forms a tube. So the name is appropriate, microtubules, so small tubes. And so, it's kind of like something like that. And each one of these is a protofilament, and I drew 13 circles. That wasn't that easy to do, but I managed to do that. And the microtubules are very interesting because they're very dynamic structures. So it turns out that the protofilaments, each individual protofilament, can either increase in length, which they call polymerization, so more monomers are added, or it can depolymerize, and the individual tubulin monomers can come off, or the Legos in the case of this example. So I'm just going to draw a microtubule. This isn't a protofilament, this is an entire microtubule, a polymerized microtubule. And as I said, there's a polarity to it, that this end of the molecule, or of the microtubule in this case, or the tubulin monomers, are different. And a great example of the function of microtubules within cells is the mitotic spindle. They form the spindle. And I'm sure everyone's seen many pictures of this because they're quite beautiful. And they have a microtubule organization center, MTOC, they like to abbreviate it, where the microtubules come together. And then, of course, these tips here attach to chromosomes during mitosis, like this. And the microtubules are directly involved in the separation of the chromosomes during this important cellular event. So it turns out that scientists realize that there's a dynamic end of the microtubule, and the dynamic end is the end that attaches to the chromosome in this particular case. So what I mean by dynamic is that in conditions that favor polymerization, this end grows extremely rapidly, and the other end grows slowly. So they like to term the very dynamic end the plus end of the microtubule, and they call the other end the minus end of the microtubules. So if conditions within the cell favor polymerization, monomers are added very quickly to the plus end, so the plus end grows quickly. The minus end grows slowly under favorable conditions for polymerization. Conversely, if the conditions within the cell promote depolymerization, the plus end shrinks rapidly, depolymerizes rapidly, and the minus end depolymerizes slowly. So the plus end is the dynamic end, and it shrinks and grows very quickly under different cellular conditions, and the minus end is the more stable end and grows and shrinks more slowly. Now it turns out that there can be conditions where the microtubule is growing at the plus end, that polymerization is occurring at this end, but that depolymerization is occurring at the minus end, and they refer to this as treadmilling. So now back in the mitotic spindle, they realize that the end near the chromosome is the plus end because it's the dynamic end, and it shrinks very rapidly, and in the process pulls the chromosome with it in this direction. And this is the more stable end, the minus end, near the microtubule organization center. And then on this side, this half of the spindle, of course, this is the plus end that's associated with the chromosome, and this is the minus end. So at some point, scientists figured out this really cool trick for studying the polarity of microtubules in different types of cells. It's called the microtubule hook assay. It turns out that if you purify tubulin from a tissue like cow brain, there's a lot of tubulin, a lot of microtubules in neurons, and you can biochemically purify tubulin to homogeneity, that is, just have a test tube of highly pure tubulin. And if you add it to cells exogenously, you just add this excess tubulin, an artifact occurs on the microtubule, and these hooks are formed. And the hooks are like this. And they form all along the length of all of the microtubules. And they figured out that if you just look at this part, hold on for one sec, let me remove this side of the spindle. So now if you use the microtubule hook assay on this spindle, and you're looking at the spindle from this direction, then you would see the microtubule, I mean the tube, so you have the individual protofilaments in here. I don't know how many I'm drawing here, that's probably not the right number, we know we need 13. But if you're looking from this direction, then the hooks go clockwise, like this. So if you're looking at this from this direction, they will go clockwise, and if they're clockwise, then you're looking at the plus end of the microtubule. If you were to see the hooks going in the other direction, that is counterclockwise, like this, then you're looking at the minus end of the microtubule. Okay, so now let's talk about the paper by Peter Baas and his colleagues. Again, the title is Polarity Orientation, so the orientation of microtubules in hippocampal neurons. So they're just trying to look at the polarity of microtubules in hippocampal neurons. And they say uniformity in the axon, so they're uniform in the axon and non-uniform in the dendrites. So we've drawn our neuron all semester long, and just a typical nerve cell like this with a cell body, of course, and the axon. And it turns out, in this case, these are neurons in culture, and they haven't synapsed yet. They're growing, and these are the dendrites. And so when neurons are growing, this axon, the end of the axon, is called the growth cone. and it's finding its way like this. So of course there are microtubules inside the axons like this, and there are microtubules in the dendrites as well, and I'm just going to draw these on the outside so we can talk about it. And the question is, what is the orientation? Where is the plus end and where is the minus end of the microtubules in the axons and the dendrites? Okay, so what do they do in this paper? Well, they're culturing rat hippocampal neurons on glass cover slips. So they remove brain tissue, hippocampus in this case, from neonatal rats, and they place them on the glass, and these neurons are able to grow with the right nutrients. In figure 1, they show a phase contrast micrograph of the hippocampal neurons. So they're using a light microscope with phase, which is a type of contrasting technique. In the image, you can see 2 hippocampal neurons in culture. You can see the 2 cell bodies, and you can see the 2 nuclei. They take the cover slip with the neuronal culture attached, and they submerge it in a solution that contains detergent. And the detergent permeabilizes the cell. That is, it pokes holes in the cell membrane. And then they add the exogenous tubulin. The tubulin can flow through these holes in the cell, find the microtubules, and bind as an artifact on the microtubules to create the microtubule hooks. So now they have a piece of glass, a cover slip, right, like this, and they have their neuron growing on it like this, or a few neurons, and they prepare this for electron microscopy, and we've talked about this before. So it gets fixed with fixatives such as glutaraldehyde and paraformaldehyde, and then it goes into stains like osmium and uranyl acetate. And then the sample's dehydrated, that is, you have to incubate the sample in alcohol, in ethanol, which removes all of the water, and then you submerge the sample in resin, in epoxy, like glue. And the glue hardens, and you end up with a block of resin, like this, surrounding the neuron. And if you drop this in liquid nitrogen, the glass will pop off. And so now you just have this block of epoxy resin, and you have your neuron like this with the growth cone right here. So this is the growth cone. Okay, now they just section this sample like this. They make a slice here, and they make a slice here at the top, and they make a slice really close to the growth cone like this, and they look at these different sections in the electron microscope. And if you look at figure 2, there's part A and B, which are just 2 different examples of the axon. So you have 2 photographs like this, and you can see microtubules like this. And they have some hooks. I mean, some only have 1 hook like that. Some have 2 hooks like I drew before. But they can look at each and every microtubule and determine the orientation of the hooks. And the hooks are all clockwise in this case. They show 2 different axons like this, and these are clockwise too. And they say you're looking at this sample from the growth cone. So your eye is here, looking in this direction from the growth cone towards the cell body, So that is to say that you're taking a section like this, right? So a thin section, and you're looking at the section from this direction in the electron microscope. And if you do that, then you see these two images with the the hooks hooking in the clockwise direction, and they say if you look at sections here, and you look at a section in the middle of the axon, or you look at a section here, or anywhere else along the length of the axon, all of the microtubules have hooks that hook in the clockwise direction. And we said earlier that if you have a microtubule and you're looking at the hooks, and the hooks hook in the clockwise direction, that you're looking at the plus end of the microtubule. So there are all of these microtubules in the axon, but all of them are oriented with the plus end here and the minus end here. So all of the microtubules, whoops, didn't like to do that, plus and minus. Every single microtubule is oriented in that direction. So an easy way to think of this is that the minus end faces the cell body, and of course the plus end faces the growth cone, or in a mature axon which has formed a synapse, the plus end is towards the presynaptic terminal of the neuron. Okay, pretty cool, right? Now they decided to look at the dendrites, and the first thing they did is they looked at the mid region, so the middle of the dendrite. So they cut a section like this, and they are viewing it from, these are growing too, so these are growth cones also at the tips of the dendrite, and they say that they're looking at it from the growth cone. So your eye is here looking at the section in this direction, and if you look at figure 3, the hooks, it turns out, are in both directions. Okay, so this is figure 2A and B, and this is figure 3. And there's two different images there, but if you look at the microtubules in figure 3 and stare at it carefully, you'll see that some of the hooks hook in the clockwise direction, which means you're looking at the plus end of those microtubules, but that some also hook in the counterclockwise direction. So those are minus end directed microtubules. So in the dendrite here, you have microtubules, and some have their plus end here and their minus end here, while others have their plus ends towards the end of the dendrite and the minus end towards the cell body. So the point is in the mid region of these dendrites, there's mixed polarity. The microtubules can face in either direction. Then, if you look at figure 4, they're talking about the distal tip. So a region here, and if you look from the growth cone, so your eye is here, and you're looking in this direction, the electron micrograph, the photograph taken with the electron microscope, shows that the hooks all hook in the clockwise direction, meaning that you're looking at the plus end of all of the microtubules at the distal tip, and then they look at a region close to the cell body. Again, the eye looking from the growth cone of the dendrite, and in that case, the microtubules are also all in the clockwise direction, like this. So that means that microtubules here at the distal tip or at the base here near the cell body all have the plus ends away from the cell body and the minus end towards the cell body. Okay, so let's just draw another image down here. We'll try to make it a little bigger. So we have a dendrite like this, and then we have our nucleus and our axon. Okay? So we have microtubules in the axon, and we said that the orientation of the microtubule is such that the plus end faces the growth cone, or the synapse, and the minus end faces the cell body. The minus end faces the cell body. In the dendrite, in this region here, the microtubules are set up so that the minus end faces the cell body. The minus end faces the cell body, and the plus end faces the tip of the dendrite. At the tip, if you look at microtubules, the microtubules are set up so that the minus end faces the cell body. The minus end faces the cell body, and the plus end faces the tip. So in these three scenarios, whether you're looking at microtubules in the axon, in any part of the axon, if you're looking at microtubules at the base of the dendrite near the cell body, or you're looking at microtubules at the distal tip, in all three scenarios, the minus end of the microtubule is towards the cell body, and the plus end of the microtubule faces away from the cell body. But interestingly enough, the microtubules that are located in the mid-region of the dendrite have mixed polarity, meaning some of the microtubules have the minus end towards the cell body, and others have the minus end facing away from the cell body. So I think this is a really cool paper, and it's fun to talk about. Okay, you guys, that's it. See you next time.